All right, so for this next section, we're going to look at how oxygen is transported in the blood. Now, oxygen is actually transported in the blood by two methods. It's either transported with hemoglobin in the red blood cells, and actually most of it is transported that way, or it's actually dissolved in the plasma. And very little is actually dissolved in the plasma. It's less than 2%. So about 3 mils of oxygen is actually dissolved in each liter of plasma, so it's not very much. So if we say that there's a total plasma volume of 3 to 5 liters, then only about 9 to 15 mils of oxygen actually can be dissolved in that plasma. So there's a limited amount of oxygen to meet the needs of resting body tissues, and that's 200 mils of oxygen per minute. That's usually required. So hemoglobin, which is a protein within all your red blood cells, allows the blood to transport 70 times more oxygen than can actually be dissolved in the plasma. So you might think that plasma would be the ideal way for transport, but it's just not possible. So the better thing is to use hemoglobin. And so what we're going to talk about next is how hemoglobin gets saturated with oxygen molecules. All right, so one other thing to point out is when oxygen is actually attached to those hemoglobin molecules, it's called oxyhemoglobin. When oxygen is not present, it's called deoxyhemoglobin. So just wanted you to be familiar with those terms. All right, so now we're going to get into this saturation. What you're going to see is what's called the oxyhemoglobin dissociation curve. So that's what this is. This is an oxyhemoglobin saturation. So how much is saturated? Is it nothing, 0%? Is it 100, everything? And then we will recognize down here, we talked about partial pressures of oxygen in millimeters of mercury. Okay, and we know that that goes, that can go up from, well, we don't see it in the blood go this low, but it could be 0 millimeters of a partial pressure of mercury all the way up to, over 105 is I think what we were looking at in those prior diagrams. But so what I want to point out to you is, if we look at the top, it reaches 98%. It never really reaches 100%. Okay, so 98% of oxygen is actually transported in the blood, bound to hemoglobin, so it reaches 98% because we know that other 2% is being circulated in the plasma. So remember, each molecule of hemoglobin can carry 4 molecules of oxygen, and that when that oxygen binds to hemoglobin, it forms oxyhemoglobin. If hemoglobin is not bound to oxygen, it's referred to as deoxyhemoglobin. So binding oxygen to hemoglobin depends on that partial pressure of oxygen in the blood. So as partial pressure of oxygen in the blood goes up, then we're going to increase our saturation. Okay. So this curve here in this figure is an oxygen hemoglobin dissociation curve, and that shows the amount of hemoglobin that is saturated with oxygen at these different partial pressure oxygen values. Now, notice this is a sigmoidal curve. It's an S-shaped curve. Has a flat part right here in this upper portion, has a very steep portion here. And what we notice is that as the, let me just go back. At high partial pressure concentrations, so this is high partial pressure of oxygen. So basically it's high concentration of oxygen. That would be inside where? The lungs, right? We would see large drops in oxygen result only in small changes in hemoglobin saturation, okay? So this would be large drops, but small changes in saturation, okay? That's called the loading portion of the curve, okay? This portion right here is called the loading portion of the curve. Basically, we're saying that the saturation stays high even with large changes in the partial pressure of oxygen, okay? So we still keep hemoglobin very saturated, and that means that the maximum amount of oxygen is bound. Why? Because where are we? We're in the lungs, okay? As this partial pressure of oxygen starts to decrease, eventually so does the hemoglobin saturation. It'll decrease as well. And that moves us into this steep portion of the curve. That coincides with the partial pressure of oxygen values typically found in the tissues. So here we're in the lungs, here we're in the tissues. So there are relatively small changes in partial pressure oxygen, but large changes in saturation. So this is a small change but a dramatic change in saturation. Okay, what this does is this enhances the unloading of oxygen to the tissues. We want oxygen to be dropped off at the tissues. Now, there are several different factors that will determine hemoglobin saturation. One of those factors is how acidic the blood is. And as the blood becomes acidic, the curve can actually shift. If the blood becomes basic, the curve can shift in a different direction. Okay, so I'm going to show you those other curves on different oxyhemoglobin dissociation curves. So let me move into my next figure. Okay, so this is an animation that you can link to through your book. I'm going to move on down to these oxyhemoglobin dissociation curves. All right, so pH, blood pH, can actually cause the curve to shift. So if the blood becomes more acidic, the dissociation curve is going to move to the right. If the blood becomes acidic, that means my pH is decreasing. Okay, if I have a decreased pH, it is acidic, and that's going to cause my oxyhemoglobin curve to shift to the right. That's called the Bohr effect. Okay, that means that more oxygen is being unloaded from the hemoglobin at the tissue level, so that's a right shift. Okay, so that's the Bohr effect. So just notice here that that acidic pH decreases the saturation. So it's enhancing the unloading of the oxygen at the level of the tissues, particularly our acidic exercising muscle. All right, so what about pH in lungs? The pH in the lungs is usually high, so the hemoglobin that's passing through the lungs has a strong affinity for oxygen. We want the lungs to be able to enhance this environment for hemoglobin to pick up oxygen, so it's encouraging a high saturation. At the tissue level, during exercise, that pH is lower. It's going to cause that oxygen to dissociate, okay, from the hemoglobin because it's becoming acidic and enhancing the supply of oxygen to those tissues. So with exercise, the ability to unload oxygen to the muscles is going to increase as the pH decreases or becomes acidic. Now, what about temperature? Blood temperature. That's going to be another variable. Blood temperature is going to affect oxygen dissociation. If you look at figure B, the right figure, there's an increased blood temperature there, and that shifts the curve to the right. So what happens to your muscles when you exercise them? They get warm, don't they? Guess what it's doing? That's going to also help unloading of oxygen to those tissues during exercise. Okay, so I hope that is making some sense. All right, so let's look at blood oxygen carrying capacity. All right, so the oxygen carrying capacity of blood is the maximal amount of oxygen that the blood can transport. That's going to depend mostly on how much hemoglobin there is in the blood. Each 100 mils of blood contains an average of 14 to 18 grams of hemoglobin, that's in men, and 12 to 16 in women. And each gram of hemoglobin can combine with about 1.34 mils of oxygen. So the oxygen carrying capacity of blood is roughly 16 to 24 mils per 100 mils of blood when the blood is fully saturated with oxygen. At rest, as the blood passes through the lungs, it is in contact with the alveolar air about 0.7 seconds. That's about how long it takes for it to go through the capillary bed. This is just enough time for the hemoglobin to become 98% saturated. Now, at high intensities of exercise, that contact time is going to be reduced, and that's going to reduce the binding of the hemoglobin to oxygen, and it'll slightly decrease the saturation, although the unique S-shaped curve kind of guards against any significant drops. But people with low hemoglobin concentrations, like people with anemia, they have reduced oxygen carrying capacities. So depending upon the severity of the condition that the people have, they will have some effect due to their cardiovascular system, which will try to compensate for that reduced blood oxygen content by increasing the cardiac output. So during activities when oxygen delivery can be limiting, such as somebody who is actively exercising, reduced blood oxygen content limits their performance. So they're going to have issues with exercise. Okay, so let's move on to carbon dioxide transport, which happens by 3 different ways. So remember, carbon dioxide is the waste product. So that is being released from the cells, from the tissues, and it needs to be gotten rid of away from the cells and away from out of the body. So there's 3 ways that carbon dioxide can be transported. One is this bicarbonate ion, and that's due to it dissociating from carbonic acid. The second one is being dissolved in plasma. And the third one is actually being bound to hemoglobin. So hemoglobin can actually bind to carbon dioxide, and that's when it's called carbaminohemoglobin. So first, let's look at bicarbonate ion. Now, the bicarbonate ion is the major way of transporting carbon dioxide. That's about 60 to 70% of how carbon dioxide is transported in the blood. Now, carbon dioxide can actually combine with water molecules and form carbonic acid. That's H2CO3. This reaction is catalyzed by carbonic anhydrase. That's found only in the red blood cells. So basically what happens is carbon dioxide diffuses into the red blood cell, it combines with water, and that carbonic anhydrase facilitates this reaction. Now, carbonic acid that gets produced, that H2CO3, is unstable, and it's going to quickly dissociate, and it forms hydrogen ions. And the other item, of course, is the bicarbonate ion, HCO3 minus, negative really. So this is the equation that we're talking about right here. So this is the bicarbonate ion that's formed. Okay. Now that hydrogen ion is actually going to bind potentially to hemoglobin. And when it binds to hemoglobin, that will help trigger the Bohr effect, because basically that's triggering acidic conditions. And so that's that right shift of the oxygen hemoglobin dissociation curve. So bicarbonate ion actually then, after this is made, diffuses out of the red blood cell and goes into the plasma. And so when that negative ion diffuses out of the red blood cell, there's going to be a chloride ion that diffuses into the red blood cell, and that's called the chloride shift, because basically we want to maintain the same charge in the cell. Now, as additional hydrogen ions form through this reaction, that actually enhances oxygen unloading at the level of the tissues. Why? Because things are becoming acidic. So hemoglobin acts as a buffer, binding and neutralizing the hydrogen ion. So it's going to basically prevent any significant changes in acidification. When this blood arrives at the lungs, where the partial pressure of CO2 is lower, the hydrogen ions and the bicarbonate ions are going to recombine, so this equation goes the opposite direction. Okay, so it's going to go in the opposite direction. And it's going to form carbonic acid, and that's going to quickly dissociate into carbon dioxide and water, because why? You're at the level of the lungs now. And so that carbon dioxide has to be put back in its original form so that it can diffuse out of the lungs. Okay? All right, so what about this dissolved CO2? So part of the CO2 that's released from the tissues gets dissolved, as we said, in the plasma, but it's really a small amount, 7 to 10%. And that dissolved carbon dioxide comes out of solution when the partial pressure of CO2 is low, as in the lungs. And that's why it readily diffuses from those pulmonary capillaries and goes into the alveoli so that it can be exhaled. Now, the other mechanism for transport for CO2 is carbaminohemoglobin. And that accounts for transport from anywhere from 20 to 33% of how CO2 is transported. So it's binding up to hemoglobin. And because that carbon dioxide binding is going to occur on a different part of the hemoglobin molecule than the oxygen binding, these two processes don't actually compete. So there's no impairment here in hemoglobin's original purpose. Now, the carbon dioxide binding does vary with regard to oxygenation of hemoglobin and the partial pressure of CO2. So basically, CO2 is released from hemoglobin when the partial pressure of CO2 is low. Basically, it's following its concentration gradient. So CO2 is readily released from the hemoglobin in the lungs because it's moving from high concentration to low concentration. So it can be exhaled through the alveoli. right, so let's move into what happens at the muscles. So we've been focused mostly on respiratory and cardiovascular systems, bringing air into the lungs. And we've been focused on transport of oxygen and carbon dioxide throughout the blood. So now we want to take a look at the delivery of oxygen from those capillary vessels to the muscle tissue itself. So the first thing we want to look at here is this arterial venous oxygen difference. So at rest, the oxygen content of that arterial blood is about 20 mils of oxygen per 100 mils of blood. So that's what's shown here in figure 712A. So that's this one. All right, so 20 mils of oxygen per 100 mils of blood. That's what you're seeing here. This decreases to 15 to 16 mils of oxygen per 100 mils in venous blood. So this difference in oxygen content between the arterial and venous blood is referred to as the arterial mixed venous oxygen difference, or A-V oxygen difference. So the term mixed venous, that's another way to say it, refers to the oxygen content of blood in the right atrium, which comes from all parts of the body, both active and inactive. So why is there this difference? The difference between this arterial and mixed venous oxygen content reflects the 4 to 5 mils of oxygen per 100 mils of blood that's taken up by the tissue. And so as this extraction increases, then the venous oxygen amount, right, is going to decrease. So we're going to have this arterial venous oxygen difference increase as a result. So as you might expect, the amount of oxygen taken up is proportional to its use for oxidative energy production. So the more oxidative energy production that's being done by the cell, the greater the amount of oxygen that's taken up. So as a rate of oxygen use is going to increase, that differential, that arterial venous oxygen difference is going to increase. And so it can actually increase to 15 to 16 mils per 100 mils of blood during maximal levels of endurance. And that's endurance exercise. That's illustrated right here. So this is that differential of 15 mils of oxygen that you can see here with intense aerobic exercise. Okay, what about transport in the muscle? So before oxygen can be used in oxidative metabolism, it's got to be transported into the muscle itself, and it's got to get to the mitochondria. And it does so by way of a molecule called myoglobin, and myo refers to muscle. So the myoglobin is similar to hemoglobin, but myoglobin is actually has a greater affinity. That means it's more attracted to the molecule. So it's more attracted to oxygen than actually hemoglobin is. And so that's what's illustrated here in this figure. It's showing you that at these partial pressures of oxygen, values that are less than 20 millimeters of mercury. Look at this, 20 millimeters of mercury. Look at how high the affinity is of oxygen for hemoglobin. Okay, so saturation with oxygen, myoglobin is quickly saturated right here at a partial pressure of 20, whereas we don't really see that until we get to about, at least 40. We start to see it approaching 80% saturation. Okay, so basically the myoglobin dissociation curve here is much steeper than the dissociation curve for hemoglobin. Now, myoglobin releases its oxygen content only under conditions in which partial pressure of oxygen is very low. And so up here at the top above here, you can see myoglobin unloading, and then you see hemoglobin unloading. And so what you'll notice there is that partial pressure of oxygen in the mitochondria of an exercising muscle may be as low as 1 millimeter of mercury. And so basically that's going to have myoglobin readily delivering that oxygen to the mitochondria. All right, so what factors influence oxygen delivery and uptake? So the rates of oxygen delivery and uptake depend upon 3 major variables. The first one is oxygen content of blood. Second is blood flow. Third is local conditions. So as we're exercising, these variables are going to be adjusting to ensure that oxygen delivery is sufficient for the active muscle. So under normal circumstances, we have hemoglobin is 98% saturated. Any reduction in the blood's normal oxygen carrying capacity is going to hinder that oxygen delivery and reduce the cellular uptake of oxygen. Also, a reduction in the partial pressure of oxygen of arterial blood would also lower the partial pressure gradient. That would limit unloading of oxygen at the tissue level. Exercise increases blood flow through the muscles. So blood flow is going to increase for oxygen flow. As more blood carries oxygen through the muscles, less oxygen must be removed from each 100 mils of blood, and therefore there's going to be an increased blood flow to improve the oxygen delivery. So if you have decreased blood flow, you have decreased opportunity to deliver that oxygen to the tissue. But exercise is going to enhance that. It's going to increase the blood flow to the muscles. What about local conditions in the muscles? During exercise, they're going to affect the oxygen delivery and uptake. If you have muscle activity that's increasing, it's also causing the environment there to become more acidic because lactate is being produced. Then there's muscle temperature. Muscles are heating up. Carbon dioxide is accumulating and increasing in concentration. All these changes are going to increase oxygen unloading from hemoglobin and facilitating and enhancing that oxygen delivery and uptake by the muscles. All right, so what about CO2 removal? So carbon dioxide simply moves out by simple diffusion. It's moving from high to low concentration in response to those partial pressure gradients. Muscles generate carbon dioxide through oxidative metabolism, just like we learned before. So that partial pressure of CO2 is going to be high compared to that capillary blood. So it's going to be basically diffusing from high concentration out of the muscle tissues to low concentration into those capillaries that are surrounding the muscles. All right, so what about the regulation of all of these processes? Regulation of your respiratory system. So we want to have a homeostatic balance to ensure that the blood partial pressure of oxygen and the blood partial pressure of carbon dioxide and our pH levels ensure a coordinated effort to maintain homeostasis among three systems essentially are involved here, right? The respiratory system, our muscular system, and our cardiovascular system. So a lot of this coordination is done by involuntary regulation of our pulmonary ventilation. Some of this is understood, some of it is still not fully understood. What we do know is this involves the respiratory muscles, and they're under the control of motor neurons, and those motor neurons are actually regulated by respiratory centers. So we have inspiratory respiratory centers and expiratory respiratory centers. And so those are located in the brainstem, and they're specifically located in the medulla oblongata, as well as the pons. So these two centers actually help establish the rate and the depth at which we breathe. So they basically send out different neuronal signals to your respiratory muscles. And of course you can hold your breath, right? So that means the cerebral cortex can override these signals because you have voluntary control if you want to, hold your breath. And then there's other input from other parts of the brain that play a role. Now, the inspiratory area of the brain is also called the dorsal respiratory group, or DRG, and it contains different cells that intrinsically fire and control the basic rhythm of ventilation. The expiratory area is kept silent or quiet during normal breathing. Why is that? That's because expiration is a passive process. It just happens. It's a passive process. It's not actively contracting muscles. But when we start to think about exercise, that expiratory area starts to actively send signals to the muscles of expiration, and then we are going to have two other brain centers that aid in the control of respiration. One of those is called the apneustic center, or apneustic area, and it actually has an excitatory effect on the inspiratory center, and that helps kind of make the activity of those inspiratory neurons longer. And then we have the pneumotaxic center, and that actually inhibits or switches off inspiration. So it's going to help regulate the inspiratory volume, so you're going to get more air in. Now, these respiratory centers don't act all by themselves. Breathing is also regulated and modified by the changes in chemical environments of the body. So there's sensitive areas in the brain that actually responds to high levels of CO2 and high levels of hydrogen ions. And we use chemoreceptors, and basically they're detecting chemicals, right? So the chemicals are CO2 and then hydrogen ions. And these chemoreceptors are actually located centrally, which means they're located in the brain. And they're stimulated by these increased concentrations of hydrogen ions in the cerebral spinal fluid that's around the brain. The blood-brain barrier is relatively impermeable to hydrogen ions and that bicarbonate, but the CO2 can actually diffuse across the blood-brain barrier. And then those central chemoreceptors are actually going to react to those increased hydrogen ions that are being produced. So this is going to stimulate then, of course, the inspiratory center, because you want to breathe that off. You want to get rid of that. That's going to stimulate the neurons to increase the rate and the depth of respiration. And that's going to allow you to remove that CO2 along with the hydrogen ions. Now, where are other chemoreceptors? They're actually located in the aortic arch, and they're also in where the two common carotid arteries actually split off as well. So these are actually sensitive areas. They're sensitive to blood changes and partial pressure of oxygen as well. So they're going to respond to really changes in the concentration of oxygen in your blood. They're going to respond to hydrogen ion concentration in your blood, and they're responding to carbon dioxide concentration in your blood. The carotid chemoreceptors are actually more sensitive to changes in hydrogen ions and partial pressures of carbon dioxide. The strongest influence found so far is actually due to the accumulation of CO2. That's the biggest driving force for helping regulate a person's breathing. And basically what happens is when the carbon dioxide levels become too high, that carbonic acid forms and quickly dissociates and gives off hydrogen ions. If the hydrogen ions accumulate, the blood becomes too acidic. So an increase in partial pressure of CO2 already will stimulate the inspiratory center to increase the respiration rate. It's not concerned with oxygen, but the goal here is to get rid of CO2 and limit any further pH changes. Okay, so what else do you have besides chemoreceptors? You have the pleura, the bronchioles, the alveoli. They have stretch receptors. So when these areas are excessively stretched, that's related to the expiratory center, and the expiratory center is going to respond by shortening the time it takes for an inspiration. And that's going to decrease the overinflation of these respiratory structures, and that's called the Herring-Brauer reflex. All right, so figure 714 is this next figure here. And that's showing you all these different centers that are involved. There are different control mechanisms that are involved in breathing. Emotional stress can cause changes. You may have noticed that as the temperature changes, it changes your breathing. But all of them are really important to help maintain appropriate levels of the blood and the tissue gases, as well as maintain a proper pH for normal cell functions. So even if there are small changes that are not controlled, they can jeopardize your health. And so if you look at this figure and you look at number 1, you're going to see the central chemoreceptors. Those central chemoreceptors, remember, they're in the brain, and they're detecting the partial pressure of CO2 and any pH changes. You also have peripheral chemoreceptors, and we said that those are located in the aortic arch and the carotid arteries, but you also will have some of those in the active muscles. So if there was conditions changing in the muscles, that would also stimulate those inspiratory centers. And then number 2, on the right-hand side there, you see the response. The response is for those external intercostal muscles in the diaphragm to contract, increase the volume of the thorax, draw air into the lungs, and let's get things moving along. As far as the expiration there at the bottom, the stimulus there is the stretch of the lungs, and that triggers the expiratory center. So basically the lungs have stretched out. That's a stimulus to say, okay, you can, expire the air now. And then number 4 down there, the response is mechanically, it's for the intercostal and the abdominal muscles to contract, cause that thoracic volume to decrease, and force the air out of the lungs. So that helps push the air out as part of an expiration. All right, so we've covered these respiratory centers. There's inspiratory and expiratory centers. Those are located in the brainstem, the medulla oblongata, and the pons. They help establish the rate and depth. In some physiology classes, we go into more detail. There's actually a third center that's actually involved here, but we won't get into that. And then, of course, I talked about the cortex being able to override that because you can hold your breath, right? We talked a little bit about those central chemoreceptors in the brain and how they affect the rate and the depth of your breathing to help get rid of CO2. And then the peripheral ones outside, obviously the central nervous system, right? That's what's meant here by peripheral. So those are chemoreceptors that are located in this aorta portion here, and also the carotid bodies. So they're actually located here where the carotid arteries split off from the aorta. They're sensitive to the partial pressures of oxygen, also the partial pressures of CO2 and hydrogen ion accumulation. And mechanoreceptors, those are just stretch receptors, and so they detect this change in basically size or shape or movement, however you want to think of it, of the thoracic cavity. And so when there's that excessive stretch, that's going to lead to a reduced depth of breathing, and that's that Herring-Brewer reflex. All right, that's it.